Good afternoon, everyone, and thank you, Daniel, for this introduction. So now I'd like to tell you a little bit about some recent work that I've been doing regarding liquid-liquid phase separation, or LLPS in short, in the context of chromatin organization in a very, very basic and preliminary term. I guess, there we go. Okay, so let's jump straight in. As you probably know, chromosomes at the megabase pair scale are generally organized into distinct compartments, which are thought to be stabilized by preferential interactions between loci in the same epigenetic state. So these interactions give rise to the spatial partitioning of the genome into euchromatic regions on the one hand, consisting mostly of actively transcribed genes, and heterochromatic regions on the other, consisting mostly of silent and unexpressed genes. Now, these interactions are generally mediated by a variety of architectural proteins, which include the prominent HP1 family, which has the ability to selectively bind to the H3K9 methylated histones, which is a heterochromatin-specific mark, through this dimerization mechanism that is depicted up here. It has been very recently shown that purified HP1 in vitro may have the ability to form these distinct spherical droplets that you can see down here, and which are observed to coalesce over relatively short timescales of the order of a second, consistent with a dynamic liquid-like form of organization. This could lead to a picture in which the compartmentalization of heterochromatin in vivo could be potentially attributed to the formation of these HP1 droplets, which are strongly reminiscent of the phase separation that you see in your everyday life when you try to mix oil and water, for instance, and into which heterochromatic loci could then tend to try to concentrate and colocalize due to this H3K9 HP1 binding mechanism. However, it is still very far from clear whether or not liquid-liquid phase separation of HP1 really does occur in vivo, and to which extent it may affect chromatin folding. The reason for that is that many of the experimental signatures of LLPS do not appear to be displayed by intranuclear HP1, but what I'll try to show you today is that this may simply be because these assays are not well suited for the complex intranuclear environment, so that these observations may actually not be inconsistent with the phase separation of HP1 in vivo. Okay, so first, let me say a few brief words about the minimal model that we use to describe LLPS, which is called the lattice gas model. So very briefly, it's a model which amounts to distributing a fixed amount of proteins on the lattice, which are simply represented as beads, and which may interact with one another with a certain effective energy, and which is evolved according to standard kinetic Monte Carlo rules. This basically gives you an extremely simple toy system, which only involves two free parameters, namely the protein-protein interaction energy J, and the protein density rho. But as I'll show you, it's actually sufficient to capture the salient features of LLPS, as far as we're concerned. So the phase diagram of the lattice model is here depicted on the left-hand side, where I'm showing you the fraction of proteins in the dense liquid phase as a function of protein density and interaction strength. So as you can see, it's typical of a first-order phase transition that is characterized by the sudden appearance of a finite-sized liquid domain in yellow here, as one raises a protein concentration above a certain critical threshold that I've here drawn in red. Now, the kinetic pathway through which this liquid phase forms is characterized by what is called coarsening dynamics, in which a lot of small droplets initially nucleate throughout the sample, as you can hopefully see on the right-hand side, and which then grow gradually through one of two mechanisms, either Oswald ripening, through which small droplets evaporate into the larger ones, as you've hopefully seen up here, or Brownian coalescence, through which droplets that are close together in space may percolate and fuse, as you might have seen down there. But we'll come back to that in a moment. Now, a last hallmark of LLPS is the so-called concentration buffering effect, which simply means that once you've nucleated a stable liquid phase, adding additional proteins into the system basically serves to grow the corresponding liquid droplets without changing the concentration of the surrounding gas phase. This means that the droplets basically act as a buffer that can absorb all of the additional proteins beyond the saturation point. And the concentration of the gas phase is actually invariant in the regime stability. So these three characteristics of phase coarsening kinetics, concentration buffering, and the fixed saturation concentration are the main experimental signatures of LLPS, which may all be qualitatively reproduced by this basic toy model. Now, before we go any further, let's briefly check how model parameters can be constrained by experimental data. So based on the in vitro studies of purified HP1, the critical concentration ρT in physiological buffer condition has been estimated to be of the order of 10 micromolars, which is basically, using our phase diagram, yields an effective protein-protein interaction energy of order kT, which is consistent with weak reversible binding interaction. Now, the model unit of length and time are also fixed by the typical HP1 molecular dimension, as well as its nuclear diffusion constant, which means that we are left for now with HP1 nuclear density as the sole free parameter of our very, very simplified model. However, up to this point, we've basically only considered pure HP1 proteins and have set aside all of their potential cross interactions, such as those with heterochromatin, for instance, which are expected to occur within the nucleus. So to take this into account, we now also introduce a basic polymer model of chromatin in which each monomer can be in one of two states, either red for heterochromatic regions, which may interact with HP1 with a certain energy, or black for active euchromatic regions, which do not interact with HP1. So for now, we'll be focusing on two simple test cases. First, an idealized stereocentric chromosome, in which all of the heterochromatin is concentrated at one extremity, and then a more realistic representation of human chromosome 19, with a detailed H3K9 distribution obtained from ChIP-seq. So starting with our simple idealized chromosome, we can draw the new phase diagram of the system as a function of our three parameters, which are now the intranuclear protein concentration and the strength of the heterotypic HP1 H3K9 interactions. My first observation is that the critical protein concentration at which the onset of LLPS occurs is generally pushed to lower densities by heterotypic interactions, which can lead to the observation of a perfectly stable liquid phase at concentrations well below that of the purified protein system. And the reason for that is that basically due to the preferential chromatin HP1 contact, heterochromatin can basically act as a substrate on which the HP1 droplets can grow, which greatly facilitates the LPS. So the observation of endogenous levels of HP1 below the critical concentration that is observed in vitro does generally not provide an argument against the presence of LLPS in the nucleus, like what you find in literature sometimes. Furthermore, if you look at the density of the surrounding gas phase, you realize that the saturation concentration is not constant anymore, but actually depends on the stoichiometry of the system. So what happens is that when you add proteins into the nucleus, you basically first start growing a droplet around heterochromatin, up to the point when the added proteins reach the H3K9 concentration, meaning that your heterochromatin is now fully encapsulated within the HP1 droplets, as you can see on the left-hand side. So beyond that point, increasing HP1 concentration further means that the extra proteins must now remain in the gas phase, since there are no more HP1 and H3K9 contacts to be gained by growing the droplet further. This means that the concentration of the gas phase is now increasing, so that concentration buffering is only partial, as you can see here. It's not constant anymore. All right, so finally, as we've seen before, it's also interesting to look at the kinetics of LLPS, which may be described as the time evolution of the mean droplet radius. So I'm showing you up here the result for the pure protein system in the absence of chromatin, where we see that at short times of the order of a second, the droplets basically grow as the square root of time, which is consistent with a regime dominated by nucleation, in which proteins condense from the supersaturated phase to form small droplets throughout the system. These droplets can then gradually grow with a much slower exponent of about 1/3, which is predicted by both theory, which is predicted by theory actually, but in both the case of Oswald ripening and Brown coalescence, up to the point where all the droplets have coalesced and you're basically left with a fully phase separated system actually. Now, if we add our idealized chromosome with one single heterochromatic domain, we observe that this coarsening behavior with a 1/3 exponent is basically completely inhibited. The reason for that is that coarsening describes large-scale rearrangements that are necessary when droplets are nucleated throughout the entirety of the system, which is not the case here because nucleation is largely limited to our single heterochromatic region, which leads to a much faster equilibration with a nucleation exponent of about 1/2. But in the more realistic case of human chromosome 19, down here, in which heterochromatin is more evenly distributed throughout the genome, the equilibration kinetics are significantly slower and characterized by transition from a 1/3 growth exponent at short times to an even slower 0.15 exponent at longer times. Now, this 0.15 value is somewhat puzzling because it's not predicted by standard theories of phase separation, but it can be simply interpreted as follows. So because HP1 droplets tend to strongly colocalize with H3K9, means that their dynamics are actually constrained by the diffusion dynamics of the heterochromatin that they encapsulate. So here we find that the heterochromatin diffuses with an anomalous exponent alpha, which is about 0.45, which is slightly lower than the 0.5 value predicted by the Rouse theory for generic polymers. So in that case, it's easy to show that the predicted growth exponent for Brown coalescence, which is limited by the rate of diffusion of your droplets, is exactly alpha over 3, which accounts for our 0.15 observed exponents. Now, interestingly, a growth exponent very close to 0.15 has been recently observed experimentally in vivo for a slightly different system of engineered correlate proteins, which tend to exclude chromatin rather than colocalize with it. So this would suggest that this 0.15 value could actually quite generally arise from the interplay between LPS and chromatin mechanics, regardless of the precise nature of chromatin-protein interactions. So to conclude briefly, I hope to have convinced you of the care that must be taken when trying to extrapolate in vitro observations of LPS to an endogenous in vivo setting, since both the stability range of the liquid phase and the equilibrium properties of the phase-separated system may be drastically altered by the presence of cross interactions, such as those between heterochromatin and HP1. These ideas have actually been developed in a much greater and much more general terms in a beautiful paper by Cliff Brangwyn, which I would strongly encourage you to check out if you're at all interested in these sort of things. And when it comes to the kinetics of phase separation, the anomalously slow 0.15 growth exponent that we observe could go some way towards explaining the microphase character of intranuclear organization, in which heterochromatin and HP1 tend to form multiple micron-sized compartments that are stable over extended periods of time, rather than a fully phase-separated system, as you would expect from standard LPS. So it could be interesting to check whether this 0.15 exponents may be corroborated by experiments based on HP1, similar to the correlate chromatin system that I've just mentioned. So with that, I would like to thank my host team here in Lyon, which is headed by our lovely chairman, Daniel, as well as our experimental collaborator, Gary Carpin at UC Berkeley, all of these people for funding, and of course, you for your attention.
Thank you, Maxime, for the presentation. So we'll start with a question from John.
Are you going to read it, or sorry?
No, John, are you here?
Yeah, okay, yeah, right. Yeah, I was curious about, is there good evidence for formation under wild type conditions of actual droplets of HP1 alpha in nuclei? I mean, is there very clear evidence of that?
At the moment, there is not, no. But
Yeah,
there
okay.
are actually papers claiming that is the other way around, that there's strong evidence against it. I don't know if you know the paper I'm referring to.
No, I was just, I wasn't sure about exactly what the implication was. Do you have an idea how large a droplet you would expect? This is very interesting, the viscoelastic. You know, I used to work on phase separation a lot.
Yes,
and,
sure.
It's very interesting, this low exponent when you have viscoelasticity playing a role. Do you have an idea what would be the saturating size that you would see in a likely experiment?
That's a very good question, and not one for which I have the answer at the moment.
Okay.
I guess it would depend on one side on the size of the heterochromatic domain, since essentially the droplets tend to colocalize with the heterochromatin, so there is a kind of effective length scale that is introduced in terms of the size of these domains. And of course, there's also an effect potentially of the stoichiometry. Yeah. So depending on how high the HP1 concentration to H3K9 is, you could potentially have competition effects between different domains of different sizes, which could lead to a very non-trivial size distribution, in my opinion.
Yeah.
So it's something.
Curious, like 100 nanometers, 200 nanometers, half a micron, you know, that kind of.
I guess it would be in that ballpark, yes, I would say, but it's difficult to answer more than that.
Yeah.
Okay, sorry, Maxime, but I think we are a bit late. So
just a quick question.
Yeah, another question from Cassie. Okay, Cassie, let's go.
Yeah, just very short. Would you predict a similar behavior for like polymerase or other proteins, even if they are active as opposed to be inactive, such as HP1?
That's another very good point. Activity is really not something we've thought about a lot at the moment, although we definitely would like to in the future. Yeah, it remains to be seen. I guess the underlying physics may be a little bit different because you will not have basically preferential domains of interactions as you do in the case of HP1 with the H3K9 methylated domains. things will probably be a little bit more distributed in the case of replication factories and things like that. But yes, it would be some ingredients that we would really like to introduce in the model at some point.
Sure, I'll stop by whenever I can.
Sure, I'd be glad to.